Hello, I am Mahshida Fuladivanda, postdoc at Torrance Center, and today I'm presenting one of our recent paper on structure connectivity guided ICA. I have no disclosure for this work. Presentation topics include introductions or motivations, proposed method, results, and conclusion. So let's start with introduction. Network is known as a set of nodes which have links with each other, and interestingly, human brain also known as a network. Brain is composed of different units or regions with specific functions. These functional units have interactions or connections with each other which made it a network. Today, neuroimaging is a powerful tool for studying brain network connectivity and regions. There are two kinds of connectivity based on the neuroimaging information. Structure connectivity, which is basically existing fiber pathways in human brain as it is shown in the picture. And the functional connectivity is based on a statistical dependency of regional activity, which these regions can be defined by independent component analysis or any other methods. Structural connectivity is estimated from diffusion MRI by performing tractography. Diffusion MRI is collected at different directions, so it has a direction information of the fibers in the human brain. For the functional connectivity, there are more kinds of data. One of them is fMRI signal, which includes brain activity over time. Now, let's have an introduction on independent component analysis, a data-driven method. Assume you have a mixture signal captured by different detectors and it contains information from independent and different sources. And what is your desire? Your desire is to find each sources. So how it is possible? Here, ICA could help to separate the mixture signals to the independent sources. So how is this going on in neuroimaging? Actually, ICA is mostly performed on fMRI data. fMRI is a mixture of different functional units activation, as you can see in the left side. Different activation signals have been mixtured and captured as an image over time that's called fMRI data. By performing ICA, we could separate this information to two kinds of data. First is the components, independent components or functional units, which are known as spatial maps. In the right picture, you can see these spatial maps. And the corresponding time series, which is the mixture matrix or mixing matrix, which they have a specific color based on their spatial maps. Actually, in the fMRI data, basically group analysis has been performed. It means that these spatial maps and these mixing matrices have been estimated per group. So they concatenate different images from different subjects, and they perform group ICA to identify spatial maps and corresponding time series over a group of subjects. And then by performing a back reconstruction strategy, you could have a subject-specific time courses and spatial maps. So the main issue with group ICA is performing group analysis, which increases potential of losing individual information. On the other hand, by back reconstruction strategy to individual level, it may affect the accuracy of the biological feature related to specific brain disorders. To solve this issue, adaptive ICA methods have been proposed, which identifies spatial maps and corresponding time courses in individual level rather than group level. There are a bunch of methods that have been worked in this area. So we are going to introduce one of the common methods in the next slide. One of the adaptive ICA method is spatial constraint ICA. The main concept in this method is using a prior spatial information to guide the procedure of estimating independent components. The prior spatial information would be a template of the components, which in this method, neuromark template has been used. Neuromark template has been created over different data sets, so it is a generalized template over different subjects, different data sets, and with healthy and disorders subjects. All this procedure have been occurred during a multi-objective procedure. There exist two objects. First, maximizing the similarity of the spatial maps to the template, and the second is maximizing the independency of the spatial map respect to a Gaussian variable. By computing individual level component, one can perform further analysis as functional connectivity, and then performing other procedure to study brain disorder or classifications or any other works. Also, you could see the main formula for this multi-objective procedure in the bottom of the slides. So our motivations till here are as follows. However, previous studies highlighted the importance of the multimodal models, especially in brain disorders such as schizophrenia, which affected brain structure and function, but still powerful models as constraint ICAs are single modality models. Besides, previous multimodality models are basically works on estimated features which are not jointly optimized to estimate the components. So here in this work, we propose a multimodal approach based on adaptive ICAs which incorporated connectivity information from two different modalities and tries to jointly optimize the component. In continue, we talk more detail of our proposed method. A structural connectivity guided spatially constrained ICA was proposed, which has three main objects to be followed simultaneously. First, finding an independent component by maximizing independency. Second, get information from prior spatial maps, the template. And the third is guiding functional connectivity by structural connectivity weights. For this purpose, we proposed a structurally weighted constraint distance between time courses, which we could explain with more details in the next slide. Here is mathematical formulations for our model. Two first objects are working on independent and spatially informed components. In the third one, we propose to minimize the distance of the time courses based on structural connectivity weights, which distance of time courses is another interpretation of the functional connectivity. It means where structural connectivity is strong, the distance of the time courses should be minimized, so the functional connectivity is maximized. In previous slides, we talked about main concepts of our proposed model. Now we first talk about our data sets, and then we go through the main steps of the model. For this work, we used FBurn dataset. It contains dMRI and fMRI images from 311 individuals, including schizophrenia and healthy subjects, and they all were pre-processed. As a first step, we need to estimate structural connectivity. In this work, a deterministic whole brain tractography was performed. Then, by merging with neuromark template, including 53 components as a brain region, individual level structure connectivity was estimated. In the following, by performing group level analysis, a group structure connectivity was determined to be used in main framework. After estimating structural connectivity in group level, to determine the individual level component, we have a multi-objective problem as shown here. It has three inputs: prior spatial information, pre-processed resting sfMRI, and group level structure connectivity, and three main objects as described before, and it can be seen below the main formulation. To solve this problem, we perform a linear weighting summation of objects with equal weights to include equal effect from different objects. By solving this problem, we have individually optimized component, and we can then perform any further process as functional connectivity estimation. At this section, we will show our results. To evaluate our model, we estimate the spatial similarity of the determined component with prior spatial maps, which are from neuromark template. For this purpose, for each component, the Pearson correlation was estimated for our proposed multimodal constrained ICA and conventional constrained ICA. In this figure, you see the different components. We have 53 components and the average correlation per component over all subjects. The red bar is our model, and the green bar is the result of the conventional constrained ICA. In overall, the similarity has been improved, which means more information was informed from spatial maps in our proposed model. A spatial map presentation of the estimated component shown in this slide. As you can see, significant differences were observed, which interpreted more integrated and focal components in proposed method respect to constraint ICA. Furthermore, we estimate functional connectivity for the component. For this purpose, we first categorized the estimated component in 7 different resting state networks, including subcortical, auditory, somatomotor, visual, cognitive control, and cerebellum. The functional connectivity between the components is based on the Pearson correlation. The Pearson correlation was used to estimate the FNC, and as you see, the FNC of our proposed method was significantly different with respect to the constraint ICA. At the end, we proposed a multimodal adaptive ICA to estimate independent components, structural and functional connectivity information was jointly incorporated during the IC estimations, and conclusionally, results showed more spatially and structurally guided independent component was determined. So, based on our result, we conclude that results suggested multimodal approach improved the performance of the ICA analysis. In these slides, we put our references, main references that we used in our model to determine the ICs. Thank you for your attention. I would be happy to hear from you for any further questions. And finally, I want to say thank you to our team in Trend Center for this work, and also the OHBM team for this program, and it was my pleasure to be in this community.